Hello, I'm pleased to welcome you to the Online Center's third webinar presentation for its 2023 series. My name is Rob Brittles. I'm a sessional professor with Thompson Rivers University. I'm also the team lead for the infrastructure program at Interior Health, and I'm a co-creator for the Online Health Center for Small Water Suppliers. I'd like to begin with a land acknowledgement. Thompson Rivers University campuses are on the traditional lands of the Tk'emlúps te Secwep, which is the Kamloops campus, and the Texalp, which is the Williams Lake campus. And they sit within the traditional and unceded territories of the Secwep. Just a little bit of background here. Thompson Rivers University, with their partner hosts, an annual webinar series and courses dedicated to provide technical information, resources, and tools to all small water system owners. We actually try to address topic areas of treatment, regulation, operation, maintenance, and water quality monitoring. These webinars are scheduled every 2 months, and they're offered for free of charge. Attendees who are EOCP certified can actually apply for the 0.1 CEUs towards your certification. A certificate of completion will be issued to the webinar participants when they can submit that to the EOCP. We have 1 hour presentations followed by half an hour of questions. And during the presentation, if you have a question, please jot them down and we'll try and get to the questions after the presentation. Now today, we're going to be talking about harmful algal blooms and what can be done. The webinar is going to introduce harmful algal blooms and what they are with regards to public health and how we treat for them. And our presenter today is Heather Larratt. Heather was the first recipient of a first-class honors BSc from environmental and biology, in environmental and biology from the University of Calgary in 1978. Thus, her 44 years of experience in source water quality and reservoir management. Along the way, her team has developed innovations in lake management and in mine reclamation. They specialize in interactions between water quality and algae. So asking casual questions about algae in social settings is ill-advised. Anyways, Heather, I'm going to turn over the presentation to you.
Okay. You can see the screen now?
Correct.
Okay, great. Thank you all. I'm very grateful for your time. I'm going to be offering you lots of tools and tips as I welcome you to the wonderful world of algae problems. This is what I plan to cover today. You can request this PowerPoint from the IHA host, and the webinar will also be posted, so please don't feel like you have to memorize everything necessarily. So why should you care about harmful algal blooms? Well, they are an escalating problem worldwide, and they are lasting longer and they're larger. We used to think that this was possibly because of improved monitoring, and that's true to a point. But NASA is also seeing that there's been an escalation recently. Some of this is attributable to climate change, but a lot of it is attributable to the way we're changing our landscapes. These figures are a little bit scary in terms of their scope and size, but the bottom line for you is that HABs will increase your operating expenses. The main 3 classes of algae are shown in this graph to the right. Diatoms will often start off the season with what we call a spring diatom bloom. They can actually be beneficial, but more on that in a bit. Green algae, which can be fairly benign, the blue-greens or cyanobacteria, which are a classic problem. These costs, by the way, that you see here, do not include healthcare costs. The main 3 classes of algae are shown in this image, and there's a great deal of difference in cell size. For example, the large filament you see with a spiraling chloroplast, one cell doesn't fit on the screen. Whereas for this dark cyanobacteria filament, if you can see my cursor, one cell is from one dark band to the next. And then these things over here are diatoms, and they're also one cell. So there's a lot of difference. The diatoms are actually made of glass. That means they have a glass case. So that makes them very, very difficult on filters. If they get onto your filter, they can permanently disable it. The green algae here obviously will not fit through any filter, and it's going to accumulate and clog the filters and cause potential taste and odor. These measurements that you see here in the red, 1, 5, and 25 microns, are for your reference in case you have filters of that size. You can see that none of these are going to fit very well. The dark filament, the cyanobacteria, are problematic. Not only do they clog filters, but they are potentially a source of cyanotoxins, and we'll talk more about that later, and also taste and odor. There's another type of algae that's important here, and it's not illustrated here, and that's the golden algae, and they make a truly awesome fishy taste and odor. So what exactly is a harmful algae bloom? By definition, that's when algae grow out of control while producing toxic or harmful effects. What's not a harmful algae bloom? Well, that's a matter of opinion, but it always involves an overgrowth of algae, but in this case does not directly cause toxicity or harmful effects, but it does create a habitat for trouble. And all algae blooms indicate an imbalance in the ecosystem, usually nutrient enrichment or modified hydrology. So how to see an algae bloom? The physical shape the bloom takes when you're standing on the shore looking at it is a reflection of its microscopic structure. For example, if we look at the top images, you see this mass of pinkish gray that has actually pushed up from the substrates underneath. Well, it's being created by the filaments you see with the red arrows to them. Whereas the one on the bottom is not cyanobacteria. This is a filamentous green algae. It's branched. And so these blobs that you see all over the lake surface here, you could pick them up with a rake as they'll want to felt together. I'm often asked the question, how many cells per milliliter is a bloom? So how bad is bad? Well, a superabundance is usually more than 2,000 cells per mil, and they can go all the way up to 100,000 cells per mil in a truly stupendous cyanobacteria bloom. These blooms occur naturally. They're not man-made, but of course we manage to accelerate them and make them worse. So what makes a bloom happen? These slides are intentionally kind of squished together so that you'll be able to see the difference between the various images. So starting at the top, when do you expect, if you're going to have a cyanobacteria bloom in a stream or another harmful algae bloom, when is that going to occur? So at the top with filamentous green algae, you're most likely to have a bloom during stable low flows, warm water, permanently wetted substrates are required, of course, no shade above the water, and low shading from turbidity, together with high phosphorus. And you expect this most, so these upper images here, in late summer. Interestingly enough, this is not dying filamentous green algae. This is a diatom bloom. And you're most likely to see those in a stream with stable cool flows. So more independent of water temperature and day length than the other algae. They also require full light, and they are affected by invertebrate grazing. Low phosphorus is often a trigger here with high nitrogen. And you see this most often in the winter and the spring. And definitely this can cause filter clogging for an intake that draws off a river. A special case is Didymo, which is short for Didymosphenia geminata, and that is a stalk-forming diatom. So these grayish-brown masses you see are actually the stalks, and there's a little diatom attached on the end. This one behaves invasively, and it forms huge masses that are truly impressive. And it'll form in the winter or summer or fall and under low phosphorus conditions. It can make a real mess, but because you get a Didymo bloom in 1 year doesn't mean that you'll get one every year following. They seem to be very intermittent. Finally, you can have cyanobacteria blooms in rivers and streams, either suspended, in this case, it's been released from a reservoir above, or attached to the substrates. Most cyanobacteria respond to high phosphorus, warm water, stable flows, and still water. Most are very low light tolerant. Some even like to help precipitate calcite. And stresses of various types will help trigger toxin production. So let's move our attention now to what can cause a harmful algae bloom in reservoirs and in lakes. Again, I've squashed the images together so that we can compare back and forth. So starting at the top with filamentous green algae, again, there's very similar conditions to what you would see in a flowing water in a lake, or sorry, a river. You'll have high phosphorus, most likely to see it in late summer, and filter clogging. In this image with the red arrow, you can see that the filamentous algae often starts growing along the substrates where there's lots of nutrients, and then it lifts to the surface as it ages. Again, like flowing water, diatoms in standing water, like cool water with minimal shade, you can actually get diatoms blooms starting at ice off. So as soon as the ice comes off, the diatom bloom takes off. And it can often look like the water is muddy, but it isn't. This muddy looking water column you see here is actually just a diatom bloom. These ones can be grazed by zooplankton, and that's very important for fish food. And they're again triggered by high nitrogen, but not necessarily phosphorus. And they absolutely have a requirement for silica because that's what they use to make the glass shells. They can be good for a lake because they consume nutrients and drop them to the bottom, but they're not good for filters, that's for sure. There's a new one that we can get in lakes, but not in flowing water, and that's a chrysophyte or golden algae bloom. Turns the water sort of a yellowish, greenish color. They can bloom in low nutrients, but it has to be high organic water or water with a lot of suspended bacteria. And that's because these algae are right on the border between plant and animal. So if there's lots of organic material available to them, they can switch off and not bother being photosynthetic at all. These guys produce a truly impressive, very strong fishy taste and odor that will persist through chlorination. Finally, cyanobacteria also respond to high phosphorus, warm water, still and shallow water. Some can be low light tolerant. They can be attached to the bottom or benthic, or they can be floating and suspended. And again, stress triggers toxin production. Late summer and fall are when you're most likely to encounter these. And the toxins can withstand some types of water treatment, but there'll be more on that in a minute. So the long and short of it is, if we add nutrients from urban and rural watersheds and climate change together, algae blooms will come. So what are the risks that we're facing aside from clogging filters? We'll talk about these sequentially now. Algae are plants, so they are photosynthetic. During the daytime, they produce oxygen, a lot of it, and they consume carbon dioxide, dissolved carbon dioxide, and that raises pH. And of course, it's during the daylight, water temperatures are likely to rise. But at night, these same plants are respiring. So they are consuming dissolved oxygen, releasing carbon dioxide, and thereby lowering pH. These can produce really, really large dissolved oxygen and pH swings. The dissolved oxygen can be really, really hard on fish. And so this fish that's dead here may not have been killed by toxins in this algae bloom. It may simply have been the dissolved oxygen swings. The fastest way to kill a fish is to run them short of oxygen. For water treatment, however, the pH swings can be giving you grief. And of course, nobody likes to see dead anything in their drinking water supply. It's not encouraging. And then cyanotoxins, we'll be spending some time on this. This is an ascending problem. It's increasing worldwide. What exactly is the issue? Well, there's a whole range of cyanobacteria. There's several thousand different species, and they also produce a wide range of cyanotoxins, and each of these species often produce more than one. So it gets complicated fast. Most of the known cyanotoxins have been identified with bottom scums that have been accidentally lifted to the surface, and acute doses are well known. So the amount that you need to consume to perish is a well-known number for most animals, including humans. Vulnerable animals are waterfowl, cattle, dogs, occasionally people. Human poisonings with fatalities are extremely rare. But there's also fatality reports for a whole range of animals that let you read here. Not all of them occur in Canada, obviously. But if we round up all of the algae that have been known to cause cyanotoxin poisonings, there's only 5 genuses that are guilty: Microcystis, Anabena, Aphanizomenon, Pantothrix, and Oscillatoria. And the top 3 account for over 80%, and they are affectionately known as Annie for Anabena, Fanny for Aphanizomenon, and Mikey for Microcystis. So Annie, Fanny, and Mikey cause most of the trouble worldwide. Maybe that'll help secure that in our memories. So what you're looking at is a satellite image of the Great Lakes. Lake Erie is the one in the center here with Huron above. Notice how the cyanobacteria blooms are swirling up from the shorelines that are either lined with urban centers or with farming, with little to no protection between the lake proper and the altered human landscape. 60% of the cyanobacteria blooms that have been tested for toxins did contain them. So not all, but the majority. So again, as I've mentioned before, cyanobacteria are most likely to produce toxins when they're stressed, such as low light, overgrowth, or the bloom itself is aging. Many cyanobacteria that produce toxins produce more than one type, and 88% of cyanotoxins co-occurred with taste and odor compounds. This to me is a huge advantage because there's a signal there. If the water smells very strange, there could in fact be cyanotoxins present. So let's look at some studies. If I gave you the luxury of the time to read this huge chart, you would see that some studies about cyanotoxin exposure and cancer are inconclusive, while others are conclusive. So what basically it's saying is there is a difference here between an acute dose in which the animal perishes and chronic low dose exposure. So what are the risks to humans from these exposure to cyanotoxin cocktails? And this diagram on the bottom gives you some idea of the many types, basic types of cyanotoxins and the organs that they attack or can impact. And interestingly enough, this diagram doesn't mention the skin and the cyanotoxins that can cause skin rashes. This just doesn't seem right to me, that cyanotoxins can be cancer promoters, as the references on the in red here, and they can be used as a chemotherapy drug, the references shown in blue. Of course, as with so many things in medicine, it's all a question of dose. As I mentioned, is this relatively new research? Initially, we had hoped that cyanotoxins did not bioaccumulate, so every step in the food chain, the dose increases. In fact, they do. Fish have a short lifespan, so they're unlikely to be killed by a cyanotoxin, but it can affect the consumers. Cyanotoxins accumulate in fish, especially the omnivores, and they're found mostly in the livers and guts, and less so in fish muscle tissue. Well, that's good news for humans, but not so good for animals that consume and rely on fish. I would personally caution against eating fish that have come from reservoirs that have a long history of cyanobacteria blooms. Certainly it's up to you, but the research is coming in that folks who rely on such a food resource are at increased risk of non-alcoholic fatty liver disease. So let's go for a slightly deeper dive into what is a cyanotoxin, the exposure routes, and some guidance from Interior Health Authority. The exposure routes, I got this diagram from some other researchers. Obviously, ingestion is problematic. You're drinking water, eating seafood, and most especially eating dietary supplements. The blue-green algae that you can get, you can actually buy these. They're legal in Canada, but not in the United States. So I don't know what that means exactly, except it's unfortunate because the cyanotoxins can be in quite a large dose if you're actually consuming these pills. You can also inhale them, and this is more concerning than it might seem. Personally, I'm not too terribly worried about sandcastles, but boating is a definite concern because any boating can produce an aerosol that is easier to inhale. Skin contact, as I mentioned before, some cyanotoxins affect the skin. And showering and bathing. That's actually one way to get an inhalation dose is with showering because you inhale the aerosol. In some cases, you're better served to shower with water that has cyanobacteria in it than you are to drink it, because when you inhale something, it's a direct link to your blood supply, whereas with drinking it, it has to go through your digestive system, and that affords some protection. There's 4 main types: the microcystins, cyanotoxins, saxitoxins, and cylindrospermopsins. But there's over 200 different types within those broad groups. And effective water treatment does depend on their chemistry. Methods used are varying, as you see in this table here. And some of these are beyond your financial reach. I recognize that. Definitely don't try to go it alone. And don't try to consume everything that you can find on the internet on cyanobacteria. It's absolutely daunting. Definitely, you can rely on the guidance that's coming from your interior health. There's 3 links provided here that I strongly recommend. So I get questions often, if you think you've got an algae bloom, how do you go about monitoring this thing? And it also was pointed out to me that not everybody recognizes the difference between phytoplankton and periphyton. So phytoplankton are the kind that free float. Literally, that's what the Greek means here, the Latin. And also periphyton means they're attached or benthic. It's not hard to sample these things, but let's just make sure we're all on the same page. First of all, you need a monitoring plan. This looks really complicated, unnecessarily so, I believe. You need to take samples, and if that you see you've got cyanobacteria that either are genetically proven or microscopically you've had sent in samples and you've got cyanobacteria that are capable of producing toxins, perhaps you've got test strips. I'll explain that in a minute. Definitely your next step is to notify your drinking water officer with Interior Health or whichever health authority you're in, and get help with messaging and your next steps. You need to build a monitoring and response plan. And I think the single biggest thing is to track all your observations and lab reports. Have a solid database, not a somewhere in Excel, because that's just not going to work well long-term and with changes in personnel. This might be one of the most useful slides in this presentation. And this is, I'm sure you might have heard of the jar and stick test for an algae bloom. If you remember, when we looked at the cyanobacteria bloom, what it actually looks like under a microscope. If you were to stick it in a jar, here's what you would notice about cyanobacteria blooms. It cannot be picked up on a stick. It'll just slide off. It looks like green or blue paint or pinkish gray, as in the case of this image. It will float on top of a jar initially. It will release a blue color, and it will smell septic or musty. There's a very distinct odor to cyanobacteria blooms, owing to geosmin and 2-MIB that they can release. This is very different than the bottom half of the screen where you see a filamentous green algae bloom. Now this can be picked up on a stick. It looks like green hair. It'll stay suspended in a jar and then slowly sink as it starts to decompose. It will not release a blue color, and it smells grassy or sometimes fishy because of all of the small aquatic invertebrates that are trapped inside the filaments when they start to die off. So the difference between a jar and a stick. So now you've decided that you may have a problem that you want to send a sample to a lab. How are you going to go about it? Well, you're triggered by either there's a strange color to the water, there's a strange smell, there's a smoking scum that you don't recognize, and unfortunately, this is a fairly reliable test. Animals and fish are getting sick or dying. Hopefully someday we'll be able to test these things in advance of such an obvious metric, but there you go. You want to sample as soon as you can after the incident, near the incident. Take reasonable precautions to fill a 500 ml or 1 liter plastic bottle. If you see different scums, please try to sample them separately, sample intakes separately, and keep samples refrigerated and cold during shipping to your lab. Please take note of floating scums, what they look like, and the odors if you notice any, and take photographs with a timestamp on them so that you remember the date year over year. If you suspected you had a real algae bloom issue, twice per week is fine to track change in a phytoplankton or floating algae bloom. How to talk about them or how to describe phytoplankton? Well, first of all, they can either appear as a haze to the water or a color to the water, or suspended blobs, filaments, or a floating mat with these colors and these smells. So turning your attention to the attached algae, how do you go about sampling them? Again, you notice an unusually thick layer of attached algae. It can be dark black or leathery or covering more than 50% of the substrates, or unfortunately, you've got animals that are sick and dying. Same thing, soon as possible. But here there's two options. When you're filling a bottle with water, you're automatically taking a quantitative sample. If you want to take a quantitative sample of periphyton, you need to sample a discrete area, as you see in the images at the bottom of the screen here. But failing that, you can always simply grab from a few different spots on a few different rocks and submit that to a lab. If you don't want to go to the protocol of taking a quantitative sample, I've given a reference here to one of the methods. Here you want to note the time since the last flushing flow or a significant change in flows. You want to note the presence of calcite, that's that crunchy, yellowy gray mineral that likes to precipitate in the summer. Presence of moss or other large plants. Please take photographs, but here once per week is fine to track change, even with a significant or potentially harmful algae bloom. Again, some descriptors: a glaze or a crust, tufts or streamers, blobs, mats, the colors that are typical for periphyton, and the smells. But for periphyton, there's also a numeric code that you can attempt to follow. Were the rocks slippery or not? Was it less than 0.5 of a millimeter thick, the harmful algae bloom layer? Were they slightly slippery, noticeably slippery, very slippery, more than 5 to 20 millimeters thick, or mostly obscured by an algae mat? So you can actually rank it. This can all form part of your metadata that goes along with in your records with lab results. So here's some potentially, this is a system that you can use, or you can reach out to your health authority to get help with this. Cyanotoxin testing can be done by a company that puts out something they call Abraxas strips. They're a discrete recipe that you follow. It's about a 5-step process, and it will tell you right away whether these test strips have detected a cyanotoxin or not. So they're very useful. It is possible that they will fail to detect a cyanotoxin, and they will only detect one type. depending on which strips you order. So they're useful, but they don't tell the whole story like everything else in biology. So here you want to collect a composite sample, water sample from 4 to 6 spots in a clean glass jar. It must be glass. It was previously wrapped with duct tape to keep the sample dark. You keep it cold and transfer it to your lab as quickly as you can, and then you follow the Abraxas recipe. If you get a positive test, you're definitely going to want to reach out to your health authority and if possible, get it verified by a certified lab. And this is harder to come by than you might think, to find a lab that's capable of doing this. And you can submit it to a taxonomy lab. But the thing with any lab is it's going to take you days to weeks to get your results back. So the Abraxas strips serve the purpose simply because they're much more immediate. And algae don't change daily, as I mentioned before. The blooms can take weeks to build and 3 to 7 days to collapse. So it's usually not necessary to sample more than once or twice weekly, even if you know you have the worst kind of harmful algae blooms, the ones that produce cyanotoxins. There's something brand new. It's not generally available yet, but we are helping to provide tests to genetic providers this year, but coming soon, hopefully. We will be able to submit samples to a genetics lab and they will verify exactly which cyanobacteria or other harmful algae are in your sample. Won't tell you how much, but it'll tell you presence or absence. What we're really hoping for in the future, though, is they'll be able to tell us whether the genes are present for cyanotoxin production. And this is really exciting because sometimes when cyanotoxin tests have been conducted, they have failed to notice or to report that there's cyanotoxins in the water. We don't know if that's because the cyanobacteria didn't switch the genes on, or there's varieties of that cyanobacteria that don't carry that gene at all. So this is coming soon to us, and it will help clarify what risks are and are not present with cyanobacteria. When you're conducting cyanobacteria sampling, it's really important to be safe. And there's a few things that are very concerning to me about this individual. And I wonder if you can spot a few before I point them out. I strongly recommend that when folks are working near water, they don't work alone, especially not if you happen to be wearing steel-toed boots. Please ensure that you've got stable footing, unlike this poor soul here, and use an appropriate sampling reach assist. Wear gloves. And don't spend a lot of time around a dense cyanobacteria bloom. Consider wearing a KN95 disposable mask when you're boating, or if you're going to spend a lot of time near a shoreline like this. In Florida, they've done testing of residents that live near waters that are prone to months-long cyanobacteria blooms, and they carry more cyanotoxin dose in their bloodstream than those that live remotely from cyanobacteria. So it's concerning. There's only 2 lakes in BC presently that our staff wear KN95 masks when they're boating, as they're capable of producing an aerosol. Please be especially careful when you see this decayed blue color, as you see a little bit in the left-hand image and more so in the right-hand image. When you see that decayed blue color, the cyanobacteria are lysing, and the presence of toxins in the water is most likely them. So first of all, we're going to look at tools for managing harmful algae blooms with water treatment, and we're going to talk about big facility treatment options first, simply because I'm hopeful that some of these will be able, with new technology, to be scaled down and available to a small water system in the future. My personal favorite is dissolved air flotation. This is because you're taking the organics, especially the cyanobacteria that are present in the water, with flocculants or polymers. You're lifting it to the top on tiny dissolved air bubbles, and then you're able to skim that load off before any future treatment proceeds. This is a very positive. Of course, it will remove everything but the cyanotoxins that are already released to the water, so the extracellular toxins. will pass by dissolved air flotation. Filtration is also a good option. Certainly it will accumulate the cyanobacteria and all other organics on the filter. Again, it tends not to deal with extracellular toxins unless your bloom lasts longer than a couple of weeks. And at that point, the critical part of the filter, which is the biofilm living around every filter particle, has been trained to recognize and consume the cyanotoxin as food, if you will. So that's a form of biodegradation, and we'll talk a little bit more about that in a minute. What you don't want to have is pressure of the cyanobacteria that are accumulated on the filter. The pressure on that can actually help break the cells, and that can, of course, increase the toxicity. So you're going to be dealing with more frequent backwashing. So, point of use. If you know that there is cyanotoxins present in the water, you can offer or suggest alternate water for those that are especially vulnerable. As always, that's the elderly, the very young, those with compromised immune systems, or some form of allergic response to a cyanotoxin. Again, granular activated carbon is very beneficial under low pressure only. It may actually protect against the cyanotoxicity, but also best to talk to your health officer and get some guidance there. What's definitely not a good idea, and this is counterintuitive, is boiling the water. Boiling the water, as you see, excuse me, in this lower image, can create cell lysis. That's fancy language for breaking the cells open and allowing the toxin to escape. The cyanotoxins will withstand boiling. Boiling does not affect them. So you can think of it in terms of the same thing as caffeine. Caffeine is also an alkaloid toxin similar to cyanotoxins. And as you know, if you boil your coffee for hours, it makes the caffeine more potent. It doesn't break it down. So same thing here. And of course, high pressure filters in point of use will actually increase cyanotoxicity to the user. So these are probably more appropriate or more likely to be in use in small facilities. So chlorination provides some benefit. Generally speaking, the dose to break down microsystems with chlorine is about double the normal dose for disinfection. So it does set you at greater risk for harmful disinfection byproducts. It doesn't break down every type of cyanotoxin, and it doesn't break down even the microsystems across a broad range of pHs. So some protection is possible with chlorination. And the same thing is true of UV. You can break down some of the cyanotoxins with UV light, but it's many orders of magnitude greater than the dose required for disabling or disinfecting pathogens. So usually it's financially not feasible. There is some evidence that could be linked up with other treatments to make it useful. So the long and the short is you could likely to get some protection from cyanotoxins from both of these, but certainly not a solution. So again, same with larger filtration plants. The most important part of the filter is the biofilm. And I am going to, if anyone speaks German, my apologies, but this very active organic layer is in German called Schmutzedecke. They were the first to coin the phrase, where basically the biofilm that lives on the filter deck is doing a vast majority of the disinfection work. And again, it's because of biodegradation of these toxins and algae materials on the filter itself. Also, PAC and GAC can actually provide food for your biofilm in times of shortage, should you have that benefit. So sand also provides a bit of protection. Theoretically, it can help degrade the toxins, but only if the water is in the filter for a matter of hours. And membrane and RO nanofiltration, these are phenomenal. But as you can imagine, if you've got an algae bloom of any type occurring in your water system, the probability that you're going to have troubles with biofouling is very significant, and you may need a pre-treatment step before you try to do filtration at such a small micron. Well, fortunately for us, these do naturally biodegrade. First of all, there is photochemical degradation. And basically, if you have bright light, these things can be degraded in a matter of hours to days, whereas in darkness, they can last for weeks to months. And the long and short of it is, if you have bright light and sediment present, you're likely to get a lot of natural biodegradation. But in the absence of these, you're going to get breakthrough of the cyanotoxins potentially through to your system. The rates that have been measured in and around the world vary depending on local conditions, as you can see in this chart at the bottom. This concludes our discussion of the treatment options, and we're going to turn our attention to trying to safeguard our source water from cyanobacteria. It goes without saying that we need the multi-barrier approach, which is always put forward by water authorities in Canada, and I'm quite sure elsewhere as well. It just makes sense. So first of all, looking at flowing water protection, if I turn your attention to this graph on the bottom, the near bed velocities, if they're very low, as you see in the early left-hand part of the graph, those are actually ideal for algae growth. These algae like having nutrients gently passed along their growing surfaces. But if you get past the point where sand starts to shear, above by 0.2 meters per second near bed velocity, then these algae start to shear off. They're actually carried away as the water velocity increases. I always think it's kind of fun that sand shears before silt, because silt likes to stay glued together. But by this point, where you're at 0.3 and 0.4 meters per second, the algae will start shearing off the substrate and be rinsed off. So obviously, initially, this is much worse for any intake on a river system, but these large velocities basically push a reset button and the algae is gone and has to start building back up again. Fall fishing flows are especially good for limiting overwinters algae blooms, which could keep oxygen low and cause off taste and odors in the winter. And summer algae accumulations can be managed by periodic flushing flows, but either you need storms at convenient intervals or you need some form of storage or reservoir so that you can release a flushing flow. If you have no sunlight, you have no algae problem, but it doesn't stop the decomposers. The bacteria, the fungi, and the yeast and so on are still functional. And consider the need, if you're going to consider a cover, that you need to have gas exchange in order to prevent the buildup of swamp gas, which is methane, hydrogen sulfide, and carbon dioxide. And so you need a breathable cover. Well, the absolute best is an intact riparian area along your stream, river, or lake. They provide numerous benefits, including habitat values and filtration. I'll explain more about that in a minute. Suspended covers or shade sails above the pond or water surface also work well. A little bit of habitat value, not much. Floating shade balls, these work well. These are incredibly expensive, by the way. And they provide very limited habitat values, you can imagine. Again, it's breathable, but it doesn't provide enough sunlight for algae growth to proceed. And one of the latest and greatest is to have floating solar panels. So it provides shade and produces electricity. As you can imagine, not much habitat value there, but it is effective. Also, if you have a constructed pond and fish haven't taken up residence in it so that it's not considered a fishery, you can drain it. End of water, end of algae problem. However, bear in mind that decomposition proceeds orders of magnitude more quickly when substrates are exposed to the air than they would if they were water covered. So when you re-wet those, there will be a surge of nutrients released back into the water column. Also bear in mind that redox sensitive metals, that's reduction in oxidation potential, metals can change their states or be liberated when you dewater a pond. Dewatering can also be used to control aquatic invasives. So let's turn our attention now to what can we do to reduce algae bloom intensity in lakes and reservoirs? What can we do to protect our source water? Again, it goes without saying, we need the multi-barrier approach. This is one approach or a first barrier among many. The issue of watershed protection will always be about preventing nutrient loading and altering the flow regime. Always try to identify the areas where you're getting high nutrient loads and work with stakeholders to develop a watershed protection plan. The OBWB toolkit is phenomenal and the link is provided here. I'm also going to provide you with a couple of our favorite tools for looking at source water protection. The first is the Google Earth time slider. Please have a look at what's happening to the forests here. This is from 1980 to present in the Okanagan. Obviously, it's not wise to think that this watershed can function the way it did back in 1980. Watch this time for the changes in the urban settings. Since we all live in houses made of wood in urban settings, you know, we are guilty of this as well. So we can't just say those awful logging people. The interesting thing is the amount of the watershed that needs to be protected here in the Okanagan to provide source protection is between 1 and 3.3% is critical to water. Obviously, we need more than that, but the critical areas are so small, I think they deserve protection. Also, if you haven't heard of it, this will be the best tip you get today is Sentinel Hub. And you can use this to see algae blooms from this satellites. This is the European Space Agency. You're looking at Wood Lake. The left-hand image is from April 14. There was a cyanobacteria bloom then, but it was dispersed through the water column, so you couldn't see it. But on April 14th, you can see these light green swirls. That's telling you, and also on April 19th. So very convenient, can form part of your permanent record. Riparian restoration cannot be overstated. This is the 1 to 3% that provides the greatest amount of protection in our water supplies. And you can go about it with planting and protecting. An exclosure is just a fenced area around a planting area that keeps it protected, because as you can see in this cross section, you get filtration and you get erosion protection from these areas. When you design riparian restoration, do think about flooding events. Finally, and this is an increasing problem, is boats are getting bigger and more powerful. What you see is a wake surf boat a few seconds after it took off in a bay with soft mud, and 3 minutes later, you can see that it provided a dose of nutrient-rich mud to the water column in this bay. We suggest that boats with onboard water tanks drain them completely before moving from one lake to another to avoid transferring aquatic invasives, and also that they operate boats dead slow if they're in water shallower than 8 meters, so that we don't get sediment disturbance. So just to recap what we've looked at so far, we've looked at what's a harmful algae bloom, how to test for it, what conditions set us up for harmful algae bloom, risks from them, monitoring, tools for water treatment, tools for lowering bloom intensity, and now for a test of sorts. So if you were to see this, would you think that this was good or bad? You can see that the boat is a ways offshore and the wake is bright green and it looks like there is flakes in the water. This one's bad. This is the infamous FANI, or Phenazamenon flossacque. I would consider wearing a mask and avoid inhaling the aerosol. So is this good or bad? This definitely can be picked up on a stick, as you can see in the person's fingers here. Well, I don't think I would join this gentleman in this bloom myself, because they can host bacteria. This type of filamentous green algae bloom can host bacteria that are not good. However, it's not a harmful algae bloom. So is this good or bad? This is tiny beads in the water. It also looks like paint. This is the infamous Mikey of our Annie, Fanny, and Mikey. So is this one good or bad? This also can't be picked up, and the wind has thrown it against the shore. This one is the Warrenachinia nigeliana, and it's also a toxin producer and a harmful algae bloom. Is this one good or bad? I've actually given you the microscope image here. And you can see it's forming this green masses along the bottom and that floats to the surface. This one's good. The nickname for this one, by the way, is water silk. Never figured that one out. And I gather there was a 1980s rock band by the name Spirogyra. Is this one good or bad? It's a bit of a giveaway that the person is wearing a mask that's doing the sampling. This one's Plantothrix. So this is just a spoiler alert. Pink is not a good color for a reservoir. There are two ways to get it that I know of. The Plantothrix, as you see here, which is very capable of producing cyanotoxins, one of the very few that is known to produce cyanotoxins at all times. And the other way is to have purple sulfur bacteria, which means that there is a ton of hydrogen sulfide in your water column, and it's just not a good place to be. In either case, I don't recommend boating in pink water. This one looks like tiny beads in the water, cannot be picked up on a stick. So this is Gliotricia, also a potential toxin producer and a harmful algae bloom. What about this one? This can be picked up on a stick. This one is guilty of absolutely smothering substrates with an overgrowth, but technically it is not often considered a harmful algae bloom. It can, however, host harmful bacteria, so please beware if you see an overgrowth of this. Is this one good or bad? It looks like paint in the water or just an off color in the water. This one also is bad. And sometimes people call this one Dolly, which, is fortunate when the long name is Dolichospermum menodonte. How about this? It's also dispersed in the water and just along the shore. Well, this one is actually pine pollen, and unless you're allergic, it's not an issue. How about this one? Again, it looks like fine beads in the water, and this is our friend Dolichospermum again. And I'm always astounded at human ingenuity, and how is it that we can come up with these crazy things, products from algae? But we do. And there's also some very excellent resources that I invite you to enjoy. And I think at this point, I'll turn it back over to Rob and we can entertain any questions.
Heather, thank you for that presentation. It was really informative. I enjoyed that. Some fantastic photos too, by the way. I'm going to turn it over to the audience to see if they have any questions. Oh, just got to grab this question 1 second. All right, so I have a person asking, are there many options for continuous online monitoring for toxins at water treatment plant, or would toxin concentrations be entirely monitored through grab samples?
That's an excellent question. There are some options for microscopic photographic continuous monitoring. They are expensive and you need a trained technician to be able to go through those photographs and visually identify. I am not familiar with any online cyanotoxin sampling at this point that would actually directly affect or sample for the toxins as opposed to the cyanobacteria. If you want to reach out to me, I can get the information on the visual systems, but they're hundreds of thousands of dollars, and you still do need a trained technician. But it does allow perhaps a little bit more of an immediate response. But honestly, you can get a lot of where you need to be by being observant and watching the water. Just standing at your lakeshore and looking at the water column, it's more useful than you might think.
Thank you. Thank you. All right, we have a question from Justin here, who, when you say diatoms are glass, could you explain a bit, please?
Absolutely. They make a beautiful case. You have to go online and Google beautiful diatoms, and I really think they're really pretty. I could be biased, but the casing that they make is literally glass. It's made of silica. So they have an absolute requirement for silica in the water. But this glass case, if you can imagine trying to get that off a filter, it can be really difficult to backwash off a filter. So diatoms are not fun for filters. If you get a bloom of them, they can produce quite a range of taste and odor. My personal favorite is wet dog, but you can also get astringent, geranium, grassy. There's a few flavors that they can produce. They don't produce toxins, not in fresh water, so they're off the hook that way. Does that answer the question?
Unless the person responds again, we won't know, but that's okay. We're gonna move on to the next question there. Are there any special precautions or extra care needed to deal with sludge produced after backwashing or filtering a source with algae? Can the dewatering sludge be treated the same for disposal, etc.?
That is a great question. If you know that you have cyanobacteria present, then I think you need to take precautions when handling the sludge. And that definitely involves a KN95. It does involve wearing gloves. And you'll need to be thinking about that. It also could mean that you cannot dispose of this material as you would normally do it in a dewatering pit because of the persistence of cyanotoxins when they're kept in the dark. So they won't last forever, but they will last for a period of time. I suggest that you would need to test the sludge for cyanotoxins before proceeding with disposal. It could be, I don't know this, but it could be that you would need special handling precautions.
Mm-hmm. Yeah, that could be true. I have a question here, Heather. We see a number of waterways these days with algal blooms. Is there an easy way to predict if a waterway is going to be subject to an algal bloom in the future?
Yes, there is. Like where I showed, when you can expect trouble in those slides in a waterway, you're most likely to have minimal trouble through the winter. Winter is kind of like a break, but once the ice is off, you can start with the diatom blooms, then proceed through the green and the cyanobacteria blooms as the season progresses. You are not likely to have, almost 100% not likely to have algae bloom issues if the water source is low in nutrients. So what we call oligotrophic. It's as we have urbanized or used agriculture without providing that protection of riparian areas between us and the water that we tend to jack up the nutrient concentrations, and then these algae blooms definitely become increasingly more likely and increasingly more problematic.
Thank you. We have another question here. If a water body is being used for recreational activity, how often should it be testing it for harmful algae?
Oh, good question. I would say if you know that this water body has a history of algae blooms, that once you start to see it, and again, there is no substitute for you who are now the educated observers to keep track of visuals, take photographs, even a Secchi disc, one of those black and white quadrant discs, so you see how far down you can see it in the water column. Those observations, once that starts to look like you're increasing your turbidity or you're increasing the water color, you're seeing a visible scum, you're getting into the June, July, August, September period, then you probably want to be testing once a week. And do keep track of this because over time you'll build a history and you'll know, okay, this lake is an upper elevation lake, so I don't see trouble in it until mid-late September when the water column overturns and more nutrients are available to the surface. But on the say valley floor, lakes and reservoirs, you could be looking at cyanobacteria blooms in July, August, perhaps into September. You know, those are just broad generalizations. It really does matter. And having a history and building that database for your water supply or the lake that's being used for recreation are indispensable.
Excellent. We have another question here. For a small conventional water system, which is filtration plus UV plus chlorination for distribution, during an algal bloom, in addition to speaking with the health authority, how should operators respond?
Well, I would not, I think respond, or as in how to act, not responding to the public, perhaps.
I'm assuming, yeah.
Definitely, if you're suspicious you've got a harmful algae bloom, whether it's cyanobacteria or other, I would do samples of your raw water and your treated water and find out what the difference is between those two. And then if there's any need to, you can go back and take samples in between the different steps in your treatment train. But basically raw versus treated, let's test that. Let's do it as a first step, but definitely start that communication with your health officer and get their advice and guidance on this as well. And start building a sampling plan from there. But as I said, you know, daily is really not necessary. Things don't change that fast unless you've had like the wind swept it towards your intake, then the next day the wind swept it away. But basically, I think you're okay with weekly. That'll probably do the job for you, raw versus treated.
Okay. We have another question here. What about septic systems that are failed but are close to the watershed? How can they affect algal blooms?
I'm sorry, what failed?
What about septic systems that are failed and I'm assuming close to the water? How can they affect algal blooms?
Well, there's other issues with septic systems that have failed close to water, but if we are just focusing on algae blooms as we are today, then the question is one of nutrients being supplied to these algae blooms, and that's definitely an issue. It's very much a concern. It's like basically like a continuous stream of nutrients going into that area, and it can produce periphyton, the attached algae blooms that will be quite focused on the area where the failing septic systems are. So there's a direct risk actually to those people that are living there and to the animals or pets that they have. If you have a lot of that on a water supply, then it can also affect the whole lake as a whole. And then you're looking at the phytoplankton or floating algae blooms as well. But the periphyton will respond almost immediately to that nutrient loading that's coming at it. And that is concerning.
Yes, interesting. I have another question here. Actually, it says, the question says, we need to do a yearly periphyton test at our backcountry hut. It has been challenging to find good instructions on how to take these samples. We are certified new operators for STP systems. Some documentation is too scientific for me to understand. Can someone advise us, maybe a contact person or link?
Okay, so
that's Petra. If you'd like to send that question directly to the Online Help Center, we can help you out with that question there. Go ahead there, Heather.
It is frustrating. There's a different recipe to follow to do a quantitative periphyton. That's where you're cleaning a known area off a bunch of rocks or sand and submitting it to a lab, because then we count cells per centimeter squared of the surface area you sampled so that you get the quantitative count. Otherwise, you're just taking a grab sample and we can tell you this is here and this is not here, but we can't tell you how much or what the density is. And because density matters when you're concerned with cyanobacteria in particular, then you have to do the quantitative sampling. And it would be nice if we all agreed on exactly how you should take those samples. But basically you have to follow the same protocol year over year to get results where you can compare year-over-year trend, which is very, very useful.
Definitely. Another question here. Would backwashing algal material into a septic system have a harmful effect on the disposal field?
Oh, no, I don't. I've never heard of that. I don't suppose it's impossible, but basically, I think your septic system, all the decomposers that are in there are going to get used to the cyanotoxin as basically just another organic molecule, and they'll eat it. I mean, most septic systems withstand caffeine, which is also an alkaloid toxin. So I don't anticipate that that would be a problem. Definitely just backwashing back to a surface water supply or somewhere else where people or animals can get into direct contact with it would be far more problematic. Just bear in mind that that is an organic load to your system, but I have personally not heard of it being harmful.
Good, thank you. And I do have another question here. Would deep water intakes help with algal problems?
I'm so glad someone asked that. The short answer is yes, but in biology, you never get a clear, clean answer like that. There are cyanobacteria that are very explicitly growing in the deep water of large lakes. For example, Okanagan Lake, for those of you familiar, is a very large, very deep fjord-style lake, and it's classically considered to be oligotrophic or nutrient poor. But there is a type of cyanobacteria called Plantothrix, which will grow deep in the water column, and it's the one that always produces cyanotoxins. And so if your intake is in that deep water column, is this little algae, I'm thinking, what is it doing down there? It's dark down there. Apparently that this is one cyanobacteria that can directly absorb organic molecules through its filaments. It doesn't have to be photosynthetic if it doesn't feel like it. And so it can actually reside deep in water columns. As long as your intake has a reasonable distance from the substrate, then really there are very few that can cause trouble like this. And it has happened here in the Okanagan. And I guess fortunately for this, this same organism produces quite a strong taste and odor. So that is what has flagged the concern to the water purveyor, that they're getting complaints of taste and odor despite the treatment train. So it's hard to have a win on this. And that system that I'm thinking of is moving towards using DAF as the first part, dissolved air flotation as the first part of their treatment train.
Well, okay. And I do have another question here. So just to clarify, if we have an algal bloom, our interior health officer can assist us in determining if it is safe at a property on a lake, and there is one section that has algae permanently. It is marshy. We'll take a shot at that for you.
Yeah, do you want to answer that, Rob?
Sure, no problem there. So yes, for the person who's asked the question here, interior health actually does address algal blooms from the perspective of if you are a permanent water supplier, you have an obligation to inform your drinking water officer, and then the drinking water officer can take steps with you to determine as to whether the algal bloom is potentially harmful, and they have the capacity to take samples. For private property homeowners, you can report any algal bloom to the Ministry of Environment through the Report All Polluters and Poachers line. That's a 24-hour hotline for a conservation officer to address the issue. That is actually also coordinated with Interior Health, and Interior Health may actually provide a general statement about the lake or lake shore or beach. It's usually directed towards beaches and private home intakes for that approach.
Good point.
I got another question. Oh, wow. This is good. Would having multiple septic systems located nearby a lake lead to more frequent algal blooms? If so, is there anything that can be done to reduce this?
Yes. How about that? Another short answer. If you have multiple septic systems on a lake, especially a small lake, So you think about the load versus the volume of the water in the lake, definitely. This can increase algae blooms of all the different types. What can be done about it? Well, one of the most obvious is that people maintain their septic systems properly and consider going to one of the more modern packaged treatment plants. Now I'm talking outside my sphere of expertise. I know they exist, and I know that if you have a disposal field close to the lake shore, if you can either move that further away from shore or, and this isn't going to be popular, plant shrub willow because it does affect your view of the lake, but something that can actually intercept those nutrients before they immigrate to the lake, that can be very beneficial. So that intact riparian vegetation layer can provide some protection also. And this is really important these days. If you've got large power boaters, encourage them to be in deep water before they power up, because as soon as they disturb the sediments are the storage area for nutrients and contaminants that come in from the watershed. That's a service that all lake sediments provide. But if we disturb those sediments, we destroy that. And we were able to actually recycle nutrients back up to the water column. So you get sort of a double whammy effect happening there. So those would be my suggestions for you.
Excellent, good stuff. I do have another question here. It says, with the treatment process of ozone, roughing filter, slow sand filter, granulated activated carbon, would shutting down the ozone help relieve the risk of release of toxins?
I'm not familiar with that being beneficial because ozone is one of the things that's used to try and address cyanotoxins. It sounds like there's quite a treatment train in place here. And again, I think perhaps if you have concerns, you know you have cyanobacteria and you're concerned and rightly so perhaps, I would suggest that you take a sample before and after your treatment system and try these Abraxas strips and see if you get a hit. If you've got it occurring before, but it's not persisting through your treatment train, then I think you're good. And if you're concerned, despite that, for some reason you have someone with immune compromised health in your household, then for the period or the duration of the bloom, you could consider bringing in an alternate water. It's probably not the answer you wanted. But I don't think dropping the ozone is really going to help you that much. I recognize that it can possibly cause cell lysis, but it also deactivates some cyanotoxins. So,
Right. The pre and post sampling is very important, though.
I think maybe that would be the best next step. And if it's not cyanobacteria at all you've got is a whole lot of organic carbon going at your filter and possible off flavors and odors. You know, to me, that's a much less serious. It still is important because people will move from a safe water supply that tastes or looks bad to an unsafe one that tastes good.
Mm-hmm.
That is true. So, you know, this is why we always consider aesthetics to be important.
Yeah, one of our watchers here has stated that there's a new resource through, I believe it's Ministry of Environment, called BC Algal Watch.
Right.
Yeah, I do believe that website helps individuals identify various algal blooms and provides some resources there for they find them.
Yeah. I really don't recommend going to the internet as a whole for your information on cyanobacteria. You will get everything from how to make products out of it to everything else under the sun. It's just simply, it's overwhelming, I think. And so going to the sources that have been presented to you here, going to your health authority, this is a great fast track through all the noise on the internet about cyanobacteria.
Yeah, I do have one more question here. Can algal blooms grow in deep wells?
No.
Or in underground aquifers?
No, absolutely not. You are off the hook. You've got to have sunlight at some point to have an algae problem. You can have bacterial problems, but they're likely to be very unrelated to cyanobacteria. You know, there'd be other carbon sources that would be more readily available to a groundwater source than say, or dissolved organic carbon emanating from an algae bloom in a surface water supply. So I would say you can just tick that one off the list. You got other issues, not this one.
Excellent. And another question here, it's in relation to copper sulfate. And is it still available or used for algal control?
Yes, it's still available. It's still not a good idea. It's an absolute last resort because copper is toxic, and there, at a certain dose, humans could take quite a lot of it. If we use copper cookware, for crying out loud, well, at least some of us do. But the dose of copper that is harmful to aquatic life is quite low. So we can't exceed that if copper sulfate is going to be used. And in order for it not to be considered a harmful substance that's going into a water supply, it has to be food grade copper sulfate, and you need a permit. And I strongly don't recommend it, because unless you're applying copper sulfate right at the very start of the bloom, then all you're doing is breaking the cells open and making the problem worse. The bloom will collapse on its own. You're just advancing that collapse. The only time that it makes any sense to use copper sulfate is if you have a full monitoring program in place and you know when you're right at the 1,000 cells per mil mark, and you're going to knock that bloom down before it can form. But I would say that 99% of the time, using copper sulfate is either not going to be allowed, or it's just like a really bad idea. And what would be a better idea is looking at what is causing this particular cyanobacteria bloom. Copper sulfate, by the way, is not particularly effective on any of the other types of algae blooms. The dose required will kill fish before you kill the algae bloom. So it's only potentially useful for cyanobacteria. And then again, like I said, it's not a good solution, and it's harmful to apply for the people that are applying it. It's not ideal. You need to be wearing some skin protection. It's just find out why you've got the bloom happening, and see if you can't cut or ratchet back the issue that's causing it, which usually is nutrients that are getting into it. There are only a couple of reservoirs I can think of. They were built over pre-existing sloughs or wetlands, and so the vast majority of their nutrient supply is internal. It's recycling within the water column itself. That's a problem. And then you might want to look at a solution such as sediment capping, where you put a thin layer of sand or some other material to isolate the nutrient source in the sediments from the water column above it. Not cheap, not easy. Try and see if there's some other way around, because honestly, copper sulfate is almost never a good idea.
Yeah. I think we're going to close there, but for anybody who has additional questions, please send them to the Online Health Center. Heather, I really want to thank you today for talking to us about harmful algal blooms. It was a really fascinating conversation. As I said, for those of you who are attending, you will receive a certificate that you can provide to the EOCP for your credits as water operators. Our next webinar is scheduled for Tuesday, June 13th from 11 till 12:30, and it's traditional and new filtration technologies for humidity removal in small water systems, and that's provided by Grant Robertson. Heather, thank you.
Hey, you're most welcome. Thank you all for your attention.
Have a good day, everybody. And one other reminder, certificates will be issued by email. You can register for the next webinar. Thank you.